go to question number 1. Lamb brush chromosomes are evident during A. Mitotic metaphase, B. Mitotic anaphase, C. Meiotic prophase, D. Mitotic prophase. Whenever we take special types of chromosomes, special type of chromosomes include lamb brush chromosomes. Lamb brush chromosomes are otherwise termed as polytene chromosomes or otherwise termed as salivary gland chromosomes. So, remember LPG, that is, they are also called as giant chromosomes. So, we have got LPGs, lamb brush, polytene, giant chromosomes or salivary gland chromosomes if they are special types found in the salivary glands of insects. Now, when you take lamb brush chromosomes, it resembles a bottle brush. So, in a lamb brush chromosome, we have a central chromatin and this one radiates on the sides like a sort of a bottle brush. So, a typical lamb brush chromosome is also analogous to a bottle brush, and it is an indication of specialized activity of the chromatin material. When you take the chromatin material could be made up of euchromatin as well as heterochromatin, functionally active euchromatin or condensed heterochromatin. Now, normally when you take lamb brush chromosomes, they are more evident especially during the time when the chromatin network is more expanded. So, the chromatin network is more expanded in the phase of meiosis, and especially in meiosis during the stage of prophase. During the phase of the prophase, there is a presence of the chromatin reticulum. Hence, lamb brush chromosomes are more significantly seen during meiotic prophase. That is when the cells are dividing and we have the prophase. The prophase is a specialized type of meiotic cell division which again can be characterized into leptotene, zygotene, pachytene, diploidine and diakinesis. And again when you take prophase, prophase can be categorized as prophase 1. So, in meiotic prophase there is the evidence of lamb brush chromosomes. Hence, among the choices the correct answer is C. We go to question number 2. Frame shift mutations is seen when A base is deleted, B, a base is added, C, when there is a shift in the reading frame, D, when anticodons are not present. We primarily understand that when you take mutations, the term mutation refers to a change in the number, kind and sequence of the nucleotides on the DNA. Supposing there is a change in the number, in the kind and the sequence of the nucleotides on the DNA which is heritable, then they are termed as mutations. Now, among mutations, we have one special type of mutation which is termed as frame shift mutation. So, in a frame shift mutation, primarily if we have the reading frame, if you think you have a reading frame called A, B and C, which forms one set of a codon. A codon is a term which is used for a sequence of 3 successive nucleotides. A set of 3 successive nucleotides like ABC constitutes a reading frame. If a reading frame like say ABC gets shifted, that is, instead of ABC it goes to form A, B, C is lost and it combines with D. Or in other words, if A and B is present and one more C is added, then BCC forms one codon, ABD forms one another sort of a codon. So if there is a shift in the reading frame always refers to 3 successive nucleotides. Whenever there is a shift, it is just like a railway engine moving on a sort of on the track. Instead of one bogie, another bogie gets attached. So primarily what happens is we have a change in the codon sequence or a change in the reading frame. So this sort of a reading frame is primarily termed as a frame shift mutation. A common example of a frame shift mutation is like if you take sickle shaped anemia. The disease which is inherited, due to which the red blood corpuscles are, instead of being turgid, they become very flaccid and they have low oxygen content because the hemoglobin content, the secondary structure of the hemoglobin content changes. So because of this you have a sickle shaped anemia. So this is a good example of a frame shift mutation. Whereas if a base is deleted or a base is added, it could primarily relate to a nitrogen base which is either an adenine, guanine, cytosine or thymine, which can get added or it can be deleted. Hence, in question number 2, the right answer is C, frame shift mutation. Question number 3, telomeres in eukaryotic chromosomes are A, cytosine rich repeats, B, guanine rich repeats, C, adenine rich repeats, D, thymine rich repeats. When you take eukaryotic chromosomes are common in the case of plants as well as in higher organisms. And in eukaryotic chromosomes, the terminal end of the chromosome is termed as a telomere. So telomeres primarily are the terminal ends of chromosomes and they are rich with relation to, primarily with relation to guanine. So they are poly G. So poly G is a characteristic feature of telomeres, especially in the case of eukaryotic chromosomes. Now what are the advantages of poly G? Supposing multiple Gs are present, n number of Gs are present, it is primarily protective in nature. The Gs prevent the chromosome from breaking, so there is no breakage, and it protects it from UV damage. As well as because of the presence of Gs, normally the chromosome will primarily anneal. That is, they will join together in case there is any sort of a broken parts. So in G there is a sort of a conservative sequence. And because of the presence of the conservative sequence, the chromosome is retained during evolution. So presence of a poly G is a characteristic feature of the telomere, and the telomere specifically takes a specific stain in chromosomal banding. In chromosomal banding, it takes a special stain wherever there is a presence of a sort of a protective G. The right answer is poly G, that is, answer is B. It is rich in guanine. Question number 4. During DNA replication, the leading strand functions in A, 5'3' direction continuously, B, 3'5' direction continuously, C, 5'3' direction discontinuously, D, 3'5' direction discontinuously. Whenever we take DNA replication, that is, supposing we have a strand of a DNA, the two strands of the DNA get separated. So when they get separated, imagine a sort of a DNA which is made up of a blue strand and a red strand, and both of them get separated. So when they get separated, there is the formation of one continuous strand on one end, and this one end continuous strand is always termed as a leading strand, and the leading strand always is always gets formed in the 5-3' direction. Always remember 3' is on the right hand side and 5' is on the left hand side. So, it gets formed from the 5' to the 3' direction and it is formed continuously. Like if we take a DNA and the DNA opens, this is unwinding. If it unwinds, at a point which is termed as the replication fork, so as it turns, one of the strands gets formed continuously. This strand which gets formed continuously, this in turn, this is called the master DNA. So, right below the master DNA, there is the formation of a continuous strand This is termed as the continuous strand. The continuous strand is formed from the 5' to the 3' direction. And the strand which is formed continuous is termed as the leading strand. So, initially a leading strand is formed just like a sort of a shadow, and after the leading strand is formed, the leading strand gets the message, and to that you have the lagging strand getting attached. So, always the most important principle here is situation number 1, we are dealing with relation to DNA replication. How does a double stranded DNA open up? 2, what does it go to form? It goes to form a leading strand and a lagging strand. What is the nature of the leading strand? It is how is it formed? It is formed continuously. And how does it get formed continuously? In which sort of direction? It gets formed in the anti-parallel direction, that is in the direction of the reverse spin, that is from 5' to 3' end. Hence, among the choices, the right answer is A, which says 5' 3' direction and continuous formation. The right answer is A. Next we go on question number 5. Triploids and monosomy is indicated as A, 2n minus 1 and 2n plus 1, B, 2n and 2n plus 1, C, 2n and 2n minus 1, D, 2n plus 1 and 2n minus 1. Whenever we are dealing with chromosomes, chromosomes can exist in the form of haploid condition, that is n, or in normal cells it can exist as a diploid condition, that is 2n. So, presence of 2n indicates that there is a union between the paternal and the maternal chromosome, resulting in a set of chromosomes which is called diploid. So, 2n is called the standard diploid situation. 2n indicates standard, that is, it is termed as a diploid condition in the case of all living organisms. Now, however, if there is an increase in a single chromosome, if we are talking in terms of single chromosomes, if there is an increase with relation to a single chromosome, here it becomes 2n plus 1. Or if there is a decrease in a single chromosome, it becomes 2n minus 1. Now, 2n plus 1 and 2s minus 1 are together termed as trisomy. Trisomy or 2n set plus one chromosome is called a trisomy, and trisomy is a characteristic feature of Datura. It is seen in Datura. And again, when you take 2n minus 1, this is termed as monosomy. Monosomy normally occurs in the case of fruits. So both trisomics and monosomics are examples of euploidy. And euploidy is a term which is used for any increase or decrease of chromosome. So under euploidy we have two situations. We have a triploid and again we have got a sort of a monosomy. Hence triploid and monosomy are indicated as 2n plus 1 and 2n minus 1, and the right answer is D. Question number 6. Salivary gland chromosomes in diphtherian larvae can be gene mapped. This is due to the nature of A. Fusion, B. Longer in size and visibility, C. Endoduplicated chromosomes, D. Easy staining. Whenever we take diphtherian larvae, in diphtherian larvae we have salivary gland chromosomes. And the salivary gland chromosomes, primarily it has got a region wherein it can puff. So puffing primarily relates to more of RNA synthesis. So when you take the RNA chromosomes, here primarily these chromosomes they show endomitosis and endomeiosis. That is, within the cell itself, one cell will keep on getting doubled. So here the chromosomes get duplicated. Now endoduplication is a very important attribute of salivary gland chromosomes of diphtherian larvae. The meaning of the term endoduplication is doubling of the chromatin material. So the density of the chromatin material as well as the quantity of the chromatin material gets doubled. Thereby it's very easy to resolute or study the sort of a resolution of the gene. Hence it is a convenient tool for gene mapping. Because of the extension of the distance between the genes. So due to the presence of endoduplication, salivary gland chromosomes are ideal tools for gene mapping. The right answer is C. Question number 7. Crossing over facilitates the ultimate function of A. Segregation of genes, B. Segregation of chromosomes, C. Recombination of alleles, D. Distribution of linked genes. When we take the process of crossing over is a mechanism of reciprocal exchange between the chromatids of two chromosomes. So when there is a reciprocal exchange during crossing over, imagine this is chromosome A and this is chromosome B. Now when chromosomes come together, this is an arm which is termed as a chromatid. So we have homologous chromosomes coming together during meiosis, and there is exchange of the genetic material. When there is exchange of the genetic material, we have two chromatids, one over here and the other one over here, and both the chromatids are non-sister chromatids. One of the chromatids belongs to A, another of the chromatids belongs to chromosome B. So there is an exchange, and this exchange of the chromatids results in the sort of a recombination there is a recombination of specifically of alleles. An allele is a term which is used for a pair of genes. Allele refers to a pair of genes, like say A and B or A dash and B dash. So, if we have A dash over here and we have B dash over here, so during crossing over A dash combines with B dash. So, there is a recombination. So, recombination of alleles is the most important objective or the ultimate function of crossing over, because crossing over is the basis for variation and it is also the basis for evolution of crop plants. Variation and evolution of crop plants. Segregation of genes means separation of genes. The objective of crossing over is not to separate or not to segregate or separate the chromosomes. The objective of crossing over, the key word here is recombination. So, recombination is a primary function of crossing over. When you say crossing over, exchange of genetic material, it means primarily genetic recombination. So, the most important thing is there is genetic recombination. There is combination between the alleles. An allele refers to a pair of genes and primarily there is exchange and the exchange results in variation and it results in the form of evolution. Hence, the answer is C. Question number 8. A second meiotic division helps in, the first meiotic division is reduction division, helps in A. Formation of 4 gametes, B. Equal distribution of haploid chromosomes, C. Equal distribution of genes, D. Segregation of the replicated chromosomes. Whenever we take meiosis, meiosis is also termed as reduction division. During reduction division of meiosis, there is reduction in the chromosome number. So, meiosis 1 deals with reduction, meiosis 1 deals with recombination, where there is genetic exchange of the material, and it is immediately followed by meiosis 2. So, during meiosis 2 there is the separation. Now, when you take the process of separation, it is also separation is also termed as segregation. When two pairs of chromosomes from the paternal and maternal chromosomes come together, there is genetic exchange of the material, there is crossing over, later on there is a formation of a tetrad and all the chromosomes get separated out. So, there is a segregation of the replicated chromosomes. The meaning of the term replicated means it is doubled. So, primarily what is the objective of meiosis 2? Meiosis 2 aims at segregation. Meiosis 2 aims at segregation. So, there is segregation of the replicated chromosomes. That is the most important function of meiosis 2. Hence, the answer is D. Question number 9. The discontinuous strand in DNA replication shows all the properties except A. The direction of growth is 5' or 3'. B. They are joined by new RNA primer and need DNA ligase. C. Formation is slower, its template opens in the 5' 3' direction. D. Okazaki fragments 3' to 5' direction of growth. When you take DNA replication, in DNA replication, after the formation of the continuous strand, this is the continuous strand. the continuous strand runs in the 5' to 3' direction. Then there is a formation of smaller fragments Now these fragments are formed in the 3' to 5' direction. They are formed from the 3' to the 5' direction. And again they are joined by new RNA primer. And RNA primer attaches to these small fragments. These small fragments are otherwise termed as Okazaki fragments. So, number 1, they are discontinuous. 2, they are termed as Okazaki fragments. 3, the direction is in the 3' 5' direction. 4th, they are joined by means of RNA primers, and in order to join they require certain other enzymes which are termed as DNA ligase. There is a formation of a DNA ligase too, and their formation is slower. While a continuous strand gets formed fast, the discontinuous strands get formed slower. And always its template, it originates from the 5' 3' direction. The continuous strand acts as a template and it moves from the 5' to 3' direction, whereas the Okazaki fragments grow in the direction of 3' to 5'. So, all the statements A, B, C and D are related to the properties of the discontinuous strand. Hence, the direction of growth in 5' to 3' direction, that is choice A is the wrong choice and it is the right answer. Answer is A. Question number 10. A gene with many expression sequences and one intron is called A. Operator gene, B. Epistatic gene, C. Split gene, D. Operon. When you take a gene, the term gene refers to a sequence of nucleotides, and when you have a sequence of nucleotides, in the sequence of the nucleotides which constitutes the gene, you require an operator gene as well as you require a regulator gene. Many times when you take genes, in genes we have sequences which express, which are termed as exons. Exons are expression sequences. That is, the DNA makes RNA, RNA makes proteins. Among the expression sequences, if you think this is an expression sequence called exon, among the expression sequences, if we have one sequence which does not express, so this is an intervening sequence. Which does not show any sort of a gene expression. So, such a combination of numerous expression sequences plus an intervening sequence is termed as a split gene. A split gene is responsible for a streak of a particular character among the most important agronomical character. So, in a split we have expression sequences with a sort of an intervening sequence, whereas an operator gene is a part of a replicon and it is a part of an operator gene, and an epistatic gene is two genes which determine a particular character, and an operon is a cluster of genes which is responsible for gene regulation. So, gene regulation, Jacobian monod and operon are associated together, whereas an expression with an intervening sequence is termed as a split gene, and the right answer is C. Question number 11. A chromosome with a terminal centromere is called A. metacentric, B. telocentric, C. acrocentric, D. submetacentric. When you take chromosomes, chromosomes are primarily the vehicles of inheritance, and chromosomes are rod-like structures which are found in the nucleus during cell division. And when you take a particular chromosome, if in a particular chromosome, if the centromere is present at the terminal end, then primarily it is termed as a telocentric chromosome. If on the same thing, if the chromosome is present in the center, it is termed as metacentric. While depending upon the position of the centromere, if the centromere is absent, it primarily termed as acrocentric. If it is absent, and again in case it is not in the middle, but it is somewhere at the sides. Now, this is the equatorial plane. If it is at the side, it is sub, that is below, so it is termed as submetacentric. So, a terminal centromere is always termed as a telocentric chromosome. The right answer is B. Question number 12. One gene, one enzyme hypothesis was postulated by A. Beadle and Totem, B. Morgan, C. Jacobian monod, D. Crick. When you take the studies of genetics, in genetics, the first persons who could postulate that a gene is responsible for a biochemical character or the link between genes and biochemistry is through enzymes. The two important Nobel laureates who had studied this hypothesis of one gene, one enzyme hypothesis were Beadle and Totem. Now, Beadle and Totem primarily worked with Neurospora crassa. While working with Neurospora crassa, they hypothesized that for every single gene is responsible for a single enzyme. That is, a DNA can make an RNA, which can make a protein, and a protein is made up of amino acids, and all enzymes are amino acids. So, the relationship between the genetic material and the biochemical component of a protein or a sort of an enzyme has been studied by Beadle and Totem. So, this was the first beginnings towards biochemical genetics. A specific gene is responsible for the synthesis of a single enzyme in a biochemical pathway means one gene, one enzyme hypothesis. A specific gene is specifically responsible for a single enzyme in the entire biochemical pathway. Later on, it has been modified as one gene, one polypeptide hypothesis, wherein it is believed that a gene, that is a stretch of a DNA with all its components, are responsible for the entire polypeptide chain which goes to form a sort of a protein. So, the fundamental belief is that a gene is responsible for a single sort of an enzyme. Hence, the answer here is A. Question number 13. Diagrammatic representation of a chromosome as per size is A. Genotype, B. Echotype, C. Ideogram, D. Karyotype. Whenever we take chromosomes and arrange them according to the sort of a sizes, this method of study is termed as karyotype analysis. When we identify chromosomes, the largest number of chromosomes of any particular organism is easily studied at metaphase. So, when we take metaphase chromosomes, take a photograph of the metaphase chromosomes and cut the chromosomes according to their sizes and arrange them on a hypothetical line with the centromeres in the center. This diagrammatic representation of the arrangement of the chromosomes according to their size is termed as karyotype. This forms the basis for counting the number of the chromosomes in any particular species and arranging the chromosomes according to the size, which is a constant. So, karyotype analysis has been done with relation to many of the sort of plants and animals, including higher organisms. And any sort of a chromosomal abnormalities like changes in the structural changes in chromosomes as well as numerical changes in chromosomes can easily be observed, or any sort of a chromosomal abnormalities like a Down syndrome or a Klinefelter syndrome can easily be observed only when you construct a sort of a diagrammatic representation of the chromosome, which is termed as karyotype analysis. Hence, the answer is D. Question number 14. Puff or Balbiani rings of salivary gland chromosomes are the sites of A. RNA synthesis, B. DNA replication, C. Protein synthesis, D. DNA duplication. Whenever we take salivary gland chromosomes, in salivary gland chromosomes, there are certain regions in the salivary gland chromosomes which can expand. They can expand primarily because there is more of chromatin material which is getting synthesized. So, any sort of a puffing or a sort of a ring which is extended and which has been discovered by a scientist whose name is Balbiani primarily represents the areas of active RNA synthesis. So, if you take the salivary gland chromosomes, there are certain regions which can primarily puff. So, these regions of puffing are active regions of RNA synthesis. So, once RNA synthesis is over, this particular puff or called, which is also termed as Balbiani rings, will again get condensed and it can just exist in the form of a sort of a band. So, this is region, this is activity region. So, after the puffing is over, it can again return to its normal higher end chromosomes. So, these regions are primarily termed as the regions of RNA synthesis. Hence, the right answer is A. Question number 15. DNA synthesis is measured by estimating the incorporation of radiolabeled A. uracil, B. thymine, C. adenine, D. deoxyribose sugar. Whenever we want to primarily study synthesis of the DNA, synthesis means how does a new strand of the DNA get formed. We have the master DNA, like you imagine DNA is double stranded. When the double stranded DNA, which is made up of a red and a blue ribbon, they separate out in the form of a red and a blue ribbon separately, the synthesis of a newer strand of a daughter DNA, the way how it primarily gets incorporated is studied by using a radio label technique involving a chemical compound called thymine. So, radiothymine is a heavy isotope. And the heavy isotope on centrifugation can settle. And because it settles, we can study by a technique called density centrifugation. And it goes to form gradients. That is, there will be a sort of a DNA which is primarily labeled with 14 thymine along with the parental DNA. So, on the basis of the gradient or based on the settling depends upon the molecular weight. So, based on the molecular weight or density centrifugation, thymine is a very important sort of a radioactive compound which is used during DNA synthesis. Hence, the answer is B. Question number 16. Neurospora crassa is a model organism because A. It can reproduce quickly and B. Easily cultured. B. Its life cycle is short. C. Mutants can be isolated that differ in nutritional requirements. And D. All the above are true. If you take Neurospora crassa, it is a fungus which has been studied by Ingold. Now, Neurospora is a haploid organism. So, when you use Neurospora for studies as a model organism, we study a branch of genetics called haploid genetics. Neurospora primarily is an organism which it sporulates. So, when you take the ascospore, this is the ascus, and within it, it has got the presence of ascospores. The ascospores are 8 in number. 8 ascospores are primarily formed from meiosis. So, the life cycle of Neurospora, it sporulates. The life cycle is short. It can easily be cultured, and we can very easily isolate mutants from Neurospora. If we have an ascospore which is a mutant, it will turn red in color, otherwise it will turn brown in color, or else it may just be half colored. So, these are all primarily mutants. So, this has formed the basis for the one gene one enzyme hypothesis of even Beadle and Totem. And you can grow Neurospora crassa, the mutants can be isolated on different medias. So, Neurospora crassa has been a very ideal organism for the one gene one enzyme hypothesis, and we can develop biochemical mutants on it. Hence, looking at the choices, choice D, that is all the above are true, is right. Hence, the answer is D.